Hello, welcome to the ecotoxicomic webinar. So today we welcome Dr. Ed Topp from Agriculture and Agri-Food Canada that will present us research about the case of antibiotic on the environment.
Merci, Chloé. Thank you, Chloé. So I'm very pleased to be here. The last time I was in Lyon was for the Ecotox conference that I think was the genesis for this network. That a number of us have been involved with since then. And so it's a great pleasure for me to give this presentation from Istria here in Lyon. So the title of my presentation is Impact of Agriculture on Environmental Microbiologisms, the case of antibiotics. My focus will be on soil microbiology, but maybe some of the things that I'll say will be applicable to aquatic microbiology, which is the interest of many of you involved in this network. So what I'm going to do is spend the first few minutes giving some very general statements about issues of sustainability and management of contaminants from primary agricultural production that can be problematic for water quality, although you'll also see that they could also be problematic for air quality. And then the majority of the presentation will focus on some experimental data that we've obtained from our research farm in London, Ontario, which is in the Great Lakes Basin in central North America, having to do with the interaction of a variety of different pharmaceuticals, particularly antibacterial pharmaceuticals, on a range of functional and molecular endpoints, essentially to ask the question, if we apply animal waste or human waste onto agricultural ground as a fertilizer, and those materials entrain pharmaceutical substances, do they do something to the microbiology in that environment that we need to worry about and therefore to manage? So agriculture is a very complex business. If you're producing a crop or a chicken or a cheese, you have to make many, many decisions, complex decisions in how you do that so that you're profitable, the food is of good quality, the food is safe, you're protecting environmental quality, animal welfare if you're in the livestock production business. But if you distill all that down to the fundamentals, it's actually fairly straightforward. So what we do in modern agriculture, if you look at the top left-hand corner, is we go into the surface of the planet and we mine phosphorus and potassium, and then we burn fossil fuels in order to convert dinitrogen gas from the atmosphere into a form that crops can use. So we now have NPK at the cost of emissions of a lot of greenhouse gases. We take that NPK, we apply it to soil, and we ask plants that have the ability to photosynthesize to fix carbon dioxide out of the air. And then we take those plants, harvest them, and we feed them to people. Typically that can be in an urban environment, for example. So we export them to urban environments that can be close by or at great distance. We take those plants and we feed them to animals, and we feed those animals to people, or the products of those animals to people. And then a portion of those nutrients are excreted by people, for example, and go into sewage treatment systems. And if the sewage treatment system is effective, the portion of the nutrients that are released directly into receiving bodies should be very low. That's what sewage treatment systems were designed for. But then a large portion of those nutrients are recovered in the form of sewage sludge. And in many jurisdictions, many systems, mine for example, the waste from people and the waste from animals are recycled back onto agricultural land. So that's essentially what we're doing in agriculture. So if you look at crop production systems or food production systems, and you think about it with respect to economic sustainability, environmental health, and human health, one way to look at it is to think about the inputs that you're putting into that production system. And those inputs fundamentally are nutrients. We talked about NPK. They're energy in the form of fossil fuel to fix nitrogen or drive a tractor or light a barn. They're water. And they're chemicals that we use to essentially kill diseases and pests that are problematic. So in crop production systems, these would be pesticides, herbicides, insecticides, fungicides. And in animal production systems, really we'd be talking about antibiotics. Antibiotics being chemicals that are used to prevent or treat infections largely with bacteria. Many would argue also that another key input is soil, because soil stocks are declining, and they're not the way that we currently practice agriculture. They are not a renewable resource. So overall, these inputs represent a cost to the producer and therefore a consumer. They may have limited availability. So, for example, we're mining, we think, all of the phosphorus that is currently available. We're concerned that we're running out of phosphorus mines, or the application may be mistimed. So, for example, rains come at inappropriate times, or water may not be available. And many of these chemicals represent both a human and an environmental health risk, which many of you work on. These would include pesticides and antibiotics. So when you think about designing agricultural production systems from the economic, human health, and environmental health sustainability perspective, what you want to do is design those systems so they're as efficient as possible, and therefore require the minimum optimal amount of inputs of these various things that we talked about. So going back to this idea of recycling nutrients from crop ground, from animals back into crop ground, from people back into crop ground, the rationale for doing that is very, very strong. And what you want is your farming systems to be as tightly coupled with your animal needs and human needs as possible. Some people might term that part of the circular economy. So that recycles and conserves nutrients and therefore represents less cost to the producer. It improves soil structure and therefore water holding capacity, for example. And at a time where we have climate variability, water holding capacity is becoming more important. It helps sequester, return carbon back into soil and maintain soil organic matter. And if one of your policy objectives is to fix more carbon dioxide out of the atmosphere into soil, it contributes to that. And at the end of the day, it improves crop yields. So again, you want to circulate CNPK from crop ground into animals, back into crop ground, and the same thing with people. But these are fecal materials, and fecal materials contain agents that are problematic for human health and the environment. So looking at this list from the top to the bottom, raw fecal material will contain enteric microorganisms, some of which may be pathogenic, viral organisms, parasitic organisms, bacteria, and some of which may be resistant to antibiotics, which will be what we'll focus on soon. These materials, in particular human waste, but also animal waste, can contain natural estrogenic or androgenic molecules that may be of concern with respect to endocrine disrupting effects, or synthetic compounds, for example, components of contraceptive pills that are excreted that end up in sewage sludge. A variety of other pharmaceuticals, personal care products may contain metals, although those tend to be controlled through regulation in high-income countries. Nitrogen and phosphorus of concern for eutrophication. And the relative amounts and types of these contaminants that one needs to think about will often vary according to the waste, whether it's livestock waste, poultry waste, or human waste. And so if you think about protecting water quality, one way to think about it is thinking about the interface between those terrestrial production systems and those aquatic systems that you want to protect. And what you want to avoid is the movement of these chemical and microbial contaminants across that interface. And just noting that the contaminants of concern may be of direct human or environmental health concern, for example, a pesticide that can kill things, or enteric pathogens that if you drink will make you sick, or a contaminant may create a concern indirectly. So for example, eutrophication leading to cyanobacterial blooms with the toxins that are produced. And so again, thinking about that, how do you manage your terrestrial production system to protect your aquatic production system? Depending on what you're looking at, there may be numerous or multiple critical control points that you may want to understand potentially to manipulate. So for example, again, focusing on antibiotic resistance, if you look at the right-hand side, lower quadrant, what you don't want is antibiotics going into the environment. You don't want bacteria that have been selected for in people or animals going into surface water that people may be exposed to or drinking water. So how do you affect that desired endpoint? If you look at the top right-hand corner, you can manage how you apply these materials. So you have appropriate buffer strips, right application, the right time, worry about antecedent moisture content. Essentially, you don't want these contaminants to move from the point of application somewhere that's undesirable. Moving to the center of the picture, you see a lagoons holding swine waste. So in many production systems, the manure is held for some period of time before application, so you can intervene at that point. Anaerobic digestion, aerobic digestion, composting. You can look at various methods that may be driven by other economic considerations, but have the benefit of making the material more benign before it's applied to the material. And finally, in the top left-hand corner, again, if the concern is antibiotic resistance, going to humans via the environment, or for that matter, via the food chain, you have a regulatory tool. You can withdraw antibiotics, or you can specify under what circumstances and what production system antibiotics are used to be represent as low a risk as possible. The lower left-hand corner is thinking about those same kind of things, but in the sewage sludge context. So we cannot obviously regulate what drugs people consume. But moving to the center of the image, these sewage sludges that are applied, again, can be manipulated through varying the redox potential, aerobic, anaerobic, composting, these kinds of things. So it's just important to think about the production system and where you can go in and intervene to, at the end of the day, protect your water quality standards. So that's one idea. Another idea is when you look at water quality at a policy relevant landscape scale or watershed scale, a number of these contaminants can have multiple sources, and it greatly complicates recommendations that you would give to a water quality manager. So looking at fecal pollution, for example, and this is in the Canadian context, you would have three sources that you would need to worry about. You'd have agriculture, so animals in the barn, animals on pasture. You have people in urban settings or rural settings where you have septic systems, and you have wildlife. And it can be mammals on the ground, or it can be avian wildlife moving in. So if you're looking at, for example, contamination of water with E. coli at a large scale, you need to be cognizant of land use in the area and maybe deploy molecular tools to try to determine whether the contaminant came from a pig or a human or a bird. So overall, at this big picture idea with respect to managing water quality, worry about looking for opportunities within whole farming systems and thinking about that terrestrial-aquatic interface, but not only focusing on that interface. And then keep in mind multiple activities within the landscape if you're working at a large landscape scale. So that's just a few general words about contaminants from agriculture and some of the thoughts that you'd want to keep in mind when looking at reducing risk. So now let's talk about antibiotics specifically. So the reason we got into this business years ago is because we were asked by some of our regulatory people to look at issues related to the use of sewage sludge in agriculture. So in Ontario, the province where I come from, we have 13 million people, and the waste from roughly 60% of those people is recycled onto agricultural ground. And so they were getting questions from the public. Are these contaminants moving? Is it safe to produce food in this ground? Do these contaminants move to crops that we're eating? Do they move to water? Are my children going to die? Are all the frogs going to die? All of these kinds of questions. So we started looking at these in detail and looked at a variety of different endpoints. But let me just describe why we think, or decided several years ago, that antibiotics in particular might be important. So what you see here is a rank list of pharmaceuticals that is in an actual sewage sludge sample that we used in our experiments. And they're rank ordered according to concentration. So the top left-hand corner, triclocarban, is the highest concentration at roughly 6 milligrams per kilogram. And the bottom right-hand corner, diazepam or Valium, is at the lowest concentration. So you see a wide variety of pharmaceuticals. Those that are highlighted in yellow are either biocidal, so they kill microorganisms generally, or antibacterial. So triclocarban is a biocide widely used in soap, for example. Ciprofloxacin is one of the fluoroquinolone antibiotics that's very sorptive and persistent in wastewater treatment systems, so it comes out in the sewage sludge. Triclosan is another biocide that's in toothpaste and other kinds of products. Another fluoroquinolone, norfloxacin, ofloxacin, Miconazole, which is an antifungal compound, tetracyclines, azithromycin, a macrolide antibiotic, et cetera, et cetera, et cetera. So when you look at this list from the perspective of a toxicologist, whether you're a microbiologist or somebody working with fish or whatever, you see a variety of agents that have different biological impacts. So the top left-hand corner, you see antimicrobials that'll kill a wide variety of microorganisms. You see different classes of antibacterial compounds, fluoroquinolones, tetracyclines, macrolides, antifungal compounds. On the right-hand side, you see other compounds that have other endpoints. Neurological endpoints, carbamazepine used to treat epilepsy and other neurological problems, different antidepressant compounds that hit different mechanisms, cardiac vascular compounds that are used to treat high blood pressure, renal compounds that are used to treat kidney problems, and finally compounds that are used to influence sterile metabolism in terms of people who have lipid problems. So a wide variety of things that are of concern. So why antibiotics? Why focus on antibiotics particularly? Well, one is we use a lot of them in agriculture, and two, the development of resistance to antibiotics is arguably one of the top 2 or 3, at least as articulated by the World Health Organization, public health problems that we currently are confronted with. So what you see here on the bottom axis is time, and what you see on the table is different classes of antibiotics that are of human health relevance. And what you see are red arrows, where the back of the arrow is the time at which that drug was introduced into use, and the tip of the arrow, the pointy tip of the arrow, is the time at which antibiotic resistance was detected. So you can see that virtually for every class of antibiotics that's been brought to market, resistance development has developed. And the problem is that the genes that are encoding resistance to these different antibiotics are accruing. They're coming together within genetic elements that are conferring upon human pathogens the ability to resist all of these different antibiotics. And in the worst case scenario, what we're very concerned with is that pathogens that are of human health concern will become resistant to every antibiotic that we have available to us. And essentially we'll go to the pre-antibiotic era where people will be dying from diseases that are currently very easy to treat, childhood diseases, very simple infections, or that medical interventions that we currently take for granted will no longer be possible. So for example, if physicians don't have access to antibiotics, they could not do transplants, a knee transplant or a pelvic transplant, because they need to use antibiotics prophylactically to prevent catastrophic infections when they cut into you. No more abdominal surgery because the risk of peritonitis is too high when you cut the gut. If you're immunocompromised and you have cancer and you're going to be treated with anti-cancer drugs that very much suppress your immune system, you're likely going to die because there won't be antibiotics to keep you from dying from infections that you're going to be infected with. So the problem with antibiotics is that more people will die in ways that we haven't seen in at least 75 years. Medical interventions that have a big impact on our quality of life will no longer be possible. And the cost to society in terms of health care will go through astronomically. So it's a big problem. So what are antibiotics? These are compounds in the word, according to the definition that I am using, compounds to kill or prevent bacteria. They have a wide range of chemical structures and therefore environmental behavior. They can be of natural products of fermentation. They can be synthetic or natural products of fermentation that are modified in some way. We see in the environment a wide range of exposure concentrations and different scenarios. So we have application of manure in a crop production context. We have irrigation with wastewater from wastewater treatment plants, and in arid parts of the world that's becoming more and more important. And we have manufacturing effluents. So factories that produce antibiotics, and I'm not familiar with the situation in France. I'm more familiar with the situation in India and China, where, as far as I know, the vast majority of drugs that we consume are produced. So around Hyderabad, for example, in India, there's a concentration of factories that produce antibiotics. And colleagues, specifically Joachim Larsson in Sweden, have looked at the concentration of antibiotics downstream from these factories. And you can get for example, ciprofloxacin, which is a fluoroquinolone, at concentrations of 30 milligrams per liter. The breakpoint concentration, the concentration that a physician would try to get in your bloodstream to kill an infection, is 4 milligrams per liter. So you have massive hotspots for resistance development in the environment because of manufacturing effluents. And then these chemicals have varied largely but not completely understood modes of action on bacteria. So for example, a number of them will inhibit protein synthesis at the level of ribosome function. So for example, the macrolide antibiotics or the tetracyclines. A number of them will interfere with the synthesis of components that are critical for cell walls, so peptidoglycan, like the penicillins, the beta-lactam antibiotics, or inhibit the biosynthesis of cofactors that are needed for various things. So for example, the folic acid that's needed for C1 metabolism, methylation of a variety of intermediates in biosynthesis, and the sulfonamide antibiotics would interact with that class of factors. So within that context, if you think about what problem we may be having on the environment that we need to understand and potentially manage, there's two areas. One is selection and enrichment of resistance of clinical concern. Are the concentrations of antibiotics that we're putting in the environment through these different exposure scenarios, are they high enough that they represent a selection pressure on environmental bacteria, increase the environmental reservoir in an undesirable way because it increases the likelihood that somehow that will end up in the clinic and be problematic for humans? And that will end up in bad things happening, as I've already described. So that's one big concern, especially within that One Health framework that we work within: agriculture, humans, the environment. And the second thing is the ecotox concern. Are we inhibiting organisms that do important things in ways that we need to worry about? So with respect to the ecotoxicological aspects of it, many people have a hard time understanding why that's important. People can understand if you're killing all the frogs or killing all the birds. I don't hear them in the morning. That's a really bad thing. But most people, including people that hand out research money, have a hard time getting their minds around the importance of microorganisms, and we don't want to screw it up. How can you illustrate that? This is what the beautiful planet looks like with all of the ecosystem services that microbiology provides. Here's what the planet looks like if we don't have those ecosystem services. So it's pretty clear. Okay, so let's talk about the agricultural context. We're applying sewage sludge, we're applying manure. We want to understand what these chemicals are doing. Are they doing any things that we need to worry about? So we've looked at 4 different areas of research. So the bottom left-hand corner, the first thing that we look at is what's the environmental behavior of these drugs? Are they persistent and therefore the window of risk is long? Or are they rapidly destroyed and therefore the window of persistence is short? Are there intermediates that accumulate that may or may not be problematic? What's the behavior of these chemicals in crop production systems that receive these wastes? The bottom right-hand corner, the second thing that we've looked at is transport of microbial and chemical contaminants to adjacent water, particularly in tiled systems. And so we've done commercial applications of these materials and asked, do they move to depth in a way that we need to worry about? The third area is on the top right-hand corner, which is, are we interfering with microorganisms in a way that we need to worry about, either through resistance or other impacts? And the fourth area is, do these contaminants move to crops that are consumed directly by humans or by animals in a way that we need to worry about? And what I'm going to do today is focus primarily on the top right-hand corner and the lower left-hand corner. I'm not going to talk about transport, and I'm not going to talk about crop contamination issues. Okay, so impact of antibiotics on microorganisms. So what's the persistence of antibiotics is a question that we have. And does long-term exposure to antibiotics promote resistance in the environmental so-called resistome? Or does it impact microbes that catalyze key ecosystem services? And we started asking ourselves this question about 20 years ago. And one of the approaches that we've used, which is the one that I'll focus on now, is to set up field experiments on our research farm in London, Ontario, and add these chemicals that we're concerned about directly to soil. The reason for that is that we can carefully control how much we're putting in. We can control the timing of the application, and we don't need to worry about the interference of the enteric microbiology with what we're seeing in the soil. So for example, if the objective is to quantify genes that confer resistance to beta-lactams, if you apply manure or sewage sludge, you don't know whether what you're quantifying came from the soil or from the fecal material that you put in. That was the rationale for designing that experiment that way. The problem with designing the experiment this way is that you don't have the fecal material that it's entrained to. So the behavior in the soil's likely to be not representative of what would happen if you applied fecal material, other kinds of considerations. So we're currently managing, I think it's roughly 140 of these small plots. And the first experiment that we started was in 1999. So we're now in our 20th year of observation in these experiments that I'll describe. So it's a very, very long-term experiment. The other set of experiments that we set up was in 2010. So we're moving into a decade of observation now. So across this suite of 140 or so plots, we have treatments that we dose at 0.1 milligram per kilogram, which is an environmentally reasonable concentration, and it's also the trigger value that regulatory authorities will go for a tiered risk assessment for pharmaceuticals that reach ground, or 10 milligrams per kilogram in the top 15 centimeters, which is arguably an environmentally excessive concentration, but it will tell us if we're going to have a treatment effect. And so the experiment that we started in 1999, we add a mixture of sulfamethazine, tylosin, and chlortetracycline, which are compounds that, at least at that time, are used in pig production. And more recently, we have plots that have received mixtures of macrolides, fluoroquinolones, cephalosporins, lincomycin and spectinomycin. So those are the antibiotic plots, or other classes of concern. So coccidiostats that are fed to chickens, antiviral compounds that are found in sewage sludge, antifungal compounds, and anti-cancer compounds that are found in sewage sludge. So every spring we go into these plots and we incorporate the drugs directly into the plots. We take samples before the incorporation, on the day of incorporation, 7 days after the incorporation, 30 days after the incorporation, and these plots are continuously cropped to soybeans, soja. So with this sampling strategy, we can see very acute short-term effects, what happens 7 days after an application, what happens a month after an application, and we can look at multiple year effects. So when we take the sample prior to that year's application, it integrates the previous 20 years of exposure or 10 years of exposure. And throughout this experiment, we freeze soil every year, every sampling point. We have a dozen -80 freezers that are full of soils from this experiment. So we can use molecular tools and go back and quantify, look at genes that we're interested in. So now let me just describe some of the information that we've obtained from these experiments as kind of lessons learned in terms of some of the things that we're interested in. So one area is, what's the persistence of these drugs, and does it vary with exposure over time to these chemicals? So accelerated biodegradation of antibiotics, which is relevant to evolution of catabolic pathways. So what you see here on the top right-hand corner is the structure of erythromycin, which is a macrolide antibiotic, which is very important in human medicine, as are the other macrolides. And what you see in the bottom right-hand corner is biodegradation potential. So this is work that we do using LCMS-MS or radio-labeled compounds, either tritiated or C14 labeled. And what you see in the bottom right-hand corner are 3 curves where we're looking at the extractable concentration of the parent compound, erythromycin. The top curve are soils, control soils that in the field were never exposed to the erythromycin. So we're seeing sort of zero order removal of the compound. The second curve in the middle of the figure are soils that were exposed for several years, I think in this experiment 6 years, to 0.1 milligram per kilogram mixture of erythromycin, clarithromycin, and azithromycin. And you can see that the compound erythromycin is removed far more rapidly. And the last curve, the one on the left-hand side, is soils that were exposed to the environmentally unreasonable 10 milligrams per kilograms in situ. So you can see that soils that have a history of exposure to this molecule at environmentally relevant concentrations of 0.1 milligram per kilogram will remove the compound more rapidly. And our argument would be what that would do is reduce environmental exposure to these antibiotics and therefore selection pressure for resistance that is of clinical concern. We haven't seen this only with the macrolide antibiotics. We've seen it with other classes of antibiotics, for example. So the bottom left-hand corner, this is sulfamethazine, the structure of sulfamethazine, which is one of the sulfonamide antibiotics. And if you look in the middle, on the right-hand side, in the middle panel, you're looking at conversion of radiolabeled sulfamethazine to carbon dioxide. And the top curve in the middle panel is rapid mineralization of the molecule by the soil that's been exposed in situ to 10 milligrams per kilogram. And you see less rapid mineralization by soils that were either not exposed to the chemical or at lower concentrations. And from these experiments, we were able to isolate a bacterium and are working with our colleagues in France on describing that bacterium, pathways, all of these kinds of things. So different classes of antibiotics can, with long-term exposure over multiple years, select for organisms that otherwise we don't see that can metabolize and grow on these molecules. As part of that, we've looked at the behavior of these drugs in situ. So if you've been applying a macrolide antibiotic to soil for 8 years, does the concentration of that antibiotic go up? And what you see in this figure here, let's look at the left-hand panel, is the concentration of erythromycin in 3 years with 3 applications. So at the left-hand side, you see 2010. We applied erythromycin, so you see the concentration of erythromycin go up and then go down. Moving towards the right, 2013 application, it goes up and then goes down. 2014, it goes up and then goes down. The right-hand side, it's the 0.1 milligram per kilogram application, the same kind of pattern. So we don't see any evidence for accumulation of these drugs in situ. So another endpoint that we've looked at is different components of the nitrogen cycle. And in this particular study, we've been looking at nitrification. And what we saw was evidence for functional redundancy. And in this, I won't go into the details because of time of this experiment, but these are plots that have received a mixture of bacitracin, ivermectin, and something else that I just can't remember. So a mixture of 3 different drugs. And we've been measuring nitrification rates by looking at consumption of ammonium, production of nitrate. And I'll just show you some molecular data looking at the relative abundance of ammonium oxidizing archaea and ammonium oxidizing bacteria. So it turns out that members of these 2 different kingdoms, the archaea and the bacteria, can oxidize ammonium as a source of energy. And what you see here on the bottom panel, lower left-hand side, is the relative ratio of those 2 groups on the day of application of the drugs in this year. On day 7, the 4 bars on the right received the high drug application of this mixture. The 4 bars on the left are either the control or receive the low drug application. Moving to the right 30 days after the application, the bars on the left are the low drug application. The bars on the right are the unreasonably high application. And you can see that the 10 milligram per kilogram is shifting that ratio. So we're seeing higher number of archaea relative to the bacteria. And so what we're saying is since we've maintained nitrification, archaea, which presumably are not affected by these antibiotics, these antibacterials, will move into that niche and confer nitrification activity in the soil. So an example of functional redundancy. Another area that we've looked at is symbiotic nitrogen fixation. And we've looked at bacteria that are associated with soybeans in the nodules and looked to see whether exposure to the drugs changes the populations that are within those nodules fixing nitrogen. And I won't show you any data, but it turns out that even exposure to the low drug concentrations will dramatically shift the communities that are within those nodules as determined using sequencing methods, different genotypes. And when we isolate those rhizobia and look to see whether the ones that we recover from nodules from the drug plots are more resistant to the antibiotics, because that would be your first hypothesis, You add drugs to soil, you kill sensitive bacteria. The ones that you detect in the nodules are resistant bacteria. That is not the case at all. So we're interpreting this to indicate that there are indirect cryptic effects of these drugs on the soil microbiome that are being translated somehow into nodules having different bacteria that are occupying the nitrogen-fixing nodules. And I think the next to last piece of science I want to talk about is focused on this concern that we're amplifying antibiotic resistance genes in soils that are exposed to these chemicals. And we've used molecular tools to quantify genes that are typically carried in cassettes that are quite mobile, and genes that we think are in the chromosomes of bacteria. And let me describe to you one of the things that we've done. So one of the approaches that we used was a so-called functional metagenomic approach. And what we did, if you look in this figure, at the top of the figure is we went into these different plots and we isolated soil. We took soil samples and we extracted large amounts of DNA from these soil samples, and we did it in a way that would yield large pieces of DNA. We didn't shear the DNA into small pieces. And then we clone these large fragments into vehicles called phosmids that are able to accommodate very large DNA fractions, very large DNA fragments of the order of 30 to 35 kb. And we created large libraries of soil DNA. We took those libraries and we put them in a strain of E. coli that is sensitive to these antibiotics. So when you plate that E. coli onto media that contains the antibiotics, the E. coli will not grow, you will not get colonies. But if you plate the library of E. coli that has your unknown DNA in it, and you plate that on medium, if the fragment that you've cloned carries a gene that confers resistance to an antibiotic and can be expressed in E. coli, that E. coli is resistant to that antibiotic and it will grow on the medium. So you'll detect a colony. So you can use that phenotypic approach to putatively identify fragments of DNA that carry genes that confer resistance to antibiotics. And so by doing that, we were able to identify fragments of DNA that confer resistance to many different classes of antibiotics. And just to make things simple here, let's just look at the bottom row on this table. So the strain, so we've just designated a fragment called macrotet A7. That's just the name for a DNA fragment. And this fragment does not increase resistance to gentamicin, which we've given the control E. coli a number of 1. However, it increases the resistance of E. coli to tetracycline, the second column, 16-fold. It increases resistance to erythromycin 4-fold, etc. If we look at the next row up, so we're talking about macromem A1, if you move to the right-hand side, it increases resistance to the carbapenem, meropenem over 1,000-fold, etc., etc., etc. And by looking through, spending a couple of years doing this, we were able to identify fragments that confer resistance to multiple classes of antibiotics. So beta-lactam antibiotics, sulfonamide antibiotics, cephalosporins, aminoglycosides, tetracyclines, etc., etc., etc. What we then did to all these different fragments was sequence them. And here's one example where we're sequencing roughly 34, 35 kb fragment and comparing the sequence that we've got, what's available in GenBank. This particular sequence conferred resistance to beta-lactam antibiotics. And if you look at the red arrow in the middle there, that is the sequence of a gene which is a penicillin binding protein that is well known to confer resistance to beta-lactams that is identical to what is already described in GenBank. So we rediscovered a number of genes that were already out there from different environments, different clinical isolates. However, in some instances, we discovered genes that nobody has ever seen before. And what I'll do is draw your attention. I'm not going to take you through this table. I'll just draw your attention to the 5th row in this table, which you see PPP. So this ORF, this open reading frame, which we've designated PPP, is unlike anything that has been seen in GenBank. And a fragment that carries that PPP gene confers resistance to macrolide antibiotics. Okay, so I'm not going to take you any more through the molecular characterization of those genes, than to say that this PPP protein, which is a polyproline protein, confers resistance to macrolide antibiotics through means that we do not understand yet. So the idea is that we're detecting genes in the environment that we haven't seen in the clinic yet. And so we're concerned that through the application of antibiotics in the environment, it makes it more likely that these cryptic genes at the end of the day will end up in the clinic. So the next question that we asked is, are we seeing these genes being amplified, these new genes that we've discovered, being amplified in these long-term plots that we've used? So what we did was, for each one of the genes that we've discovered, we designed quantitative PCR systems, went back into the soils, and asked, do these genes become more abundant in response to the antibiotic treatment? Because if they do, that means that they have some function in situ, and it means that exposing soils to these antibiotics is amplifying them in a way that we might need to worry about. And so I'll just show you some quantitative PCR data to make those points. So let's look at the top left-hand panel here. So this is DNA isolated from plots that have received mixtures of antibiotics for 6 years when these samples were taken. And the axis on the left-hand side is the relative copy number of these gene targets referenced to total 16S RNA, to total bacteria. And we have 3 bars for each sampling date. The white bar, the clear bar, is the untreated control that has not received antibiotics. The gray bar is the soil that has received the environmentally relevant concentration, and the black bar is the soil that has received the unrealistic 10 milligram per kilogram concentration. So the day 0 on the left-hand side, the day 0 data is integrating the previous 6 years of exposure to this drug. And you can see that this gene target, INT1, which is an integrase, which is associated with the mobility of gene cassettes, is greatly amplified in those soils that have received the excessive exposure, but not the environmentally relevant exposure. You can see that 7 days after exposure to the drug, the INT1 relative abundance goes up. So the bacteria are responding within a week to application of these drugs. And you can see a month later that it's gone down a bit. So the period at which bacteria are responding to exposure is within a few days. And it's integrating or accumulating over the period of many years. The other gene targets that you see in this figure, STRA, SIL1, STRB, and SIL2, are typically carried within these gene cassettes. And they all respond to excessive concentrations, but not environmentally relevant concentrations. This is from the same experiment. Different gene targets. These are some of the ones that we've discovered. And based on flanking sequence, we think that all these genes are within the chromosome of bacteria. And you can see that in general, none of them is responding to environmentally relevant concentrations, with the possible exception on the lower left-hand panel of this PPP gene, the gene that we've discovered that confers resistance to macrolide antibiotics. And you can see on day 30 in this sampling year, that gene is amplified significantly higher than control plots. And with the even higher exposure, it's amplified more. So we're seeing a dose response. So what's the significance of those findings? New antibiotic resistance genes in soils conferring resistance to important antibiotics, in some cases by unknown mechanisms. An increase in abundance of these novel antibiotic resistance genes in response to environmentally meaningful concentrations suggests that they're functionally important in situ, and that antibiotic exposure is promoting resistance in agricultural soils. So this is an area that we need to look at in far more detail to really nail down risk. So I think this is close to my next to last slide. And the point of this slide is to say that we have an opportunity using these long-term experiments to look at a variety of methods that people who do ecotoxicology would typically employ to look at impacts. And so if you look at the column on the left-hand side, what I've listed here is a number of endpoints that we have deployed in this experiment. We or our collaborators have deployed. So we've looked at accelerated biodegradation of sulfamethazine and other compounds, and we've seen accelerated biodegradation. And moving to the right in that column, The data that we've looked at was in the 10th year of this particular experiment. We did not see an effect at low environmentally relevant concentrations. We did see an effect at the 10 milligram per kilogram exposure, and we saw it short term in terms of a response within 7 days, and we saw it long term in terms of a response integrating previous year's activity. Looking the second row, crop residue degradation with radiolabeled material. We do not see an effect at low concentration. We do at intermediate concentration. We do not at high concentration. And moving through the table, we've looked at degradation of 2,4-D, quantifying total bacteria by PCR, quantifying SOL1, SOL2 genes that are carried by cassettes, the relative abundance of nitrifying archaea in bacteria, the RISA method that's used to look at gross composition, the molecular probes, live, dead, fish kits, fish gross composition, looking at gross group, metagenomic methods using CPN60, PLFRA, gross fungal composition, metagenomic inventories using 16S, and PICT using incorporation of radiolabeled leucine into soil bacteria. And if you look at this list, kind of the conclusion that I'm moving to is that gross methods, PLFA, RISA, PICT, do not detect treatment effects, whereas some of the very fine molecular methods like metagenomic inventories, CPN60 are detecting effects. So we're still kind of you could see that we've been working on this for many years, but I'm hoping that by the end of some point when I get around to writing it up, from these kinds of experiments, we can make some comment on what kind of test a regulatory agency would use when assessing impacts of these drugs on soil microbiology or other things. I would say the other interesting thing about this is you would probably argue that from the aquatic perspective, a number of these tools might be effective, but they are not in the soil terrestrial context. So this is just my thank you note focused on a whole variety of people that have been involved one way or the other in the antibiotic field experiments over many years. And with that, I'm finished. Merci, thank you.